Hello everyone. The next topic which we will be discussing is about the carbon fixation. So carbon fixation is also known as the Calvin cycle and it is also known as the C3 cycle. So we will be discussing the mechanism of the carbon fixation, that what are the steps that are involved in the carbon fixation cycle, and all of these we will be discussing in detail in these slides. So let's start with the first slide.
Slide of the Calvin cycle, which is the introduction slide. Calvin cycle basically is present in all the oxygenic photosynthetic organism, that is plants. C2 reduction in fixation occurs through the carbon cycle, that is carbon fixation cycle, which is also called as the Calvin cycle. So the Calvin cycle is a series of biochemical reactions which reduce CO2 to 3 carbon sugar. So it is being converted to 3 carbon sugar, that is why it is also known as C3 cycle. In eukaryotes, it takes place in the stroma of the chloroplast, and in case of the prokaryotes, it is taken place it occurs in the cytosol. So this is the introduction part, that in eukaryotes it takes place in the stroma of chloroplast, and in case of prokaryotes it occurs in the cytosol.
So next part I will talk about is the history of the carbon fixation cycle. So basically the carbon fixation cycle, it was being discovered or elucidated by the Melvin Calvin and Andy Bonson at the University of California, Berkeley, for which they were awarded the Nobel Prize also in the year 1961. So the carbon fixation cycle, as I have already told you, is also known as the Calvin cycle. So the scientist who discovered this cycle was named after him also. So that is why it is known as the Calvin cycle or carbon fixation cycle. The next is that what are the three phases of the carbon fixation cycle. So the three phases of the carbon fixation cycle will include the carboxylation, which is the first step. The second step includes the reduction, and the third step will include the regeneration part. So we will be understanding whole of this mechanism in detail, uh, starting from the carboxylation till the regeneration part.
So carbon fixation, the first stage involves the incorporating carbon from carbon dioxide into an organic molecule. So here RUBP, the full form of RUBP is here, that ribulose 1,5-bis phosphate, which is a 5 carbon acceptor molecule. So the carbon dioxide combines to this RUBP, and an enzyme called RUBP carboxylase or oxygenase or Rubisco catalase, attachment of carbon dioxide to RUBP. So this process make a 6 carbon compound. So earlier it was a 5 carbon compound. So when an enzyme acts on this, they will make a 6 carbon compound. The compound is very unstable, so it would split into 2 to 3 carbon molecules compounds, which is 3-phosphoglyceric acid. Then hence for each of the carbon dioxide that would enter the cycle, there will be 3 PGA molecules which are formed. We will be discussing all these in the cycle in detail also. The next step which comes is the reduction step. So in case of reduction, this is the next phase. So the reduction stage requires ATP and NADPH. These compounds are used to convert the 3 PGA molecules into a 3 carbon sugar, which is known as glyceraldehyde 3-phosphate or G3P. In the first step, each molecule of 3 PGA would receive phosphate group from the ATP, and which is turning into 1,3-bisphosphoglycerate, which is a double phosphorylated molecule. So this would leave the ADP as a byproduct. So the second step which comes into this picture is the 1,3-bisphosphoglycerate molecule. They are reduced by gaining the electrons. So that is being reduced by gaining the electrons. So each of the molecules would receive 2 electrons from NADH, and they loses one of its phosphate group. So after which the glyceraldehyde 3 phosphate or G3P, a 3 carbon compound is being produced. So in the second stage of reduction, it produces phosphate and NAD plus as a byproducts. So this was about the reduction stage. Then comes the regeneration stage. What happens is regeneration is some G3P molecules, they produce glucose, while the others would recycle in order to generate the RUBP acceptors. So RUBP, in the first slide of carboxylation, uh, we have seen that what is RUBP acceptors. Then this stage would require ATP. So this stage would require the energy, and they involve a very complex set of reactions in this. So this the regeneration stage has been there. So the 3 molecules of carbon dioxide, they enter the cycle. So in order for one G3P molecule to leave the cycle and on to the glucose synthesis, and they provide the 3 new atoms of fixed carbon. So there are 6 G3P molecules which will be produced when the 3 carbon molecules will enter the cycle. So the cycle will be represented covering all these steps in detail. So one would leave the cycle to be used to produce glucose, while the rest would be recycled in order to regenerate 3 molecules of the RUBP acceptors. This is about the Calvin cycle. So here it is a picture which is being depicted here, that how a C3 or a Calvin cycle is being shown. So there are basically 4 steps in the Calvin cycle. So first step is carbon fixation. We have discussed the carbon fixation. It requires 6 CO2 molecules which will convert 12 molecules of 3 PGA, that is phosphoglycerate. The 12 ATP will be required in order to produce 12 ADP, and then the 12 NADPH molecules will be required to convert the 12 NAD plus ions. Then it will convert into 12 G3P. Then this was about the second step. Then about the third step. In the third step, what is happening? Rubisco molecule, that is 2G3P, is transported from the chloroplast to make the glucose, which gets converted into 6 ribulose, 5 phosphate, and after this they will require the ATP for this energy, and they will release the 6 ADP molecules. This is the fourth step which we are talking about, and 6 ribulose 1,5 bisphosphate molecules are being produced here. So this is about the Calvin cycle, in which carboxylation is the fixation of the carbon by the carboxylation of RUBP. Reduction is the reduction of the fixed carbon to begin the synthesis of triose phosphate, and regeneration, it is the synthesis of the starting compound RUBP. So all the steps are being covered here, all the phases are being covered here. So if we take the example, for example, if we have to write one equation on this. So what we will write is 3 CO2 molecules plus 6 NADPH molecules plus 9 ATP will give rise to G3P plus 6 NADP plus ions and 9 ADP molecules. So this is about the Calvin cycle. Then the regulation of enzymes which is involved in the Rubisco regeneration, that is the enzymes that is involved in RUBP regeneration is fructose biphosphatase and pseudohepatulase biphosphate. They are more active at the pH of 8 and at the pH of 7 something, and they require magnesium as a cofactor for catalysis. So These are two enzymes which is required for this RUBP regeneration, and they are more active at pH 8 and requires magnesium for the as a cofactor for the catalysis. They are sensitive to the redox potential of the chloroplast stromal compartments, and the mediator molecule is the small protein thyroredoxine. So thyroredoxine is when coupled with ferrodoxin uh, by the enzyme, that it will lead to the inhibition of the phosphofructokinase. So in plant cells, light stimulates the Calvin cycle enzymes. So this is the main point here. The light activation is being mediated by the thiodoxin. So this was all about the RUBP and its regeneration, the control of the Calvin cycle. This was all about this. So these are some of the references from where I have taken the matter and from where I have made the slides. Thank you.